We appreciate everybody that's come on board here for our annual Zoom cancer research update by the face of our cancer research, Dr. Annette Khaled, and give a little bit of history here and what to expect today. Dr. Khaled has been working with us. The Orlando Sports Foundation actually getting ready to celebrate 15-year birthday here on May 28th. And I know that soon after that, Dr. Annette Khaled joined us, and we, as a part of our advisory board, obviously our science board, and we've been supporting her research at the UCF College of Medicine ever since. We're closing in on a total of hopefully here by the end of Cure Cup or by our birth date on May 28th, we'll be able to announce $4 million. We're closing in on $4 million total that has been donated to cancer research. A bulk of that has been to the Breast Cancer Research Foundation and the Florida Breast Cancer Foundation. And through those organizations, Dr. Khaled received over $1.2 million of research for her work at the UCF College of Medicine. And now the UCF Cancer Research Center. Annette, congratulations. The old Sanford Burnham building has been turned into a cancer research center. And Annette has also got a new title as director of the UCF Cancer Research Center. All this is happening during our time and our work together with Dr. Khaled and the Orlando Sports Foundation and the Cure Bowl. So we're excited to have Dr. Khaled update us as we do annually on our Zoom in March. And Annette always gives us a quick update at our kickoff luncheon in August as well. And so you're certainly welcome to join us for our kickoff luncheon and meet Dr. Khaled in person. So Annette, I will turn it over to you, and we're all looking forward to hearing the updates. Thank you.
Well, thank you, Alan, and it's a pleasure. And as I said, a lot happening here at UCF as we build our new cancer center. And I'm not quite director yet, but we're working on it. So let me tell you what we've done this year. I was telling Alan that this has been an exceptionally amazing year for us in the lab. So I'm going to share my screen and get started on this presentation that I put together. And I just checking with you all. If there are any questions, are we using chat or just people can just jump in? I'll give the people time to, between slides to jump in with questions. So I think we can probably go either way, right? Okay. All right then. So we're all here because cancer is something that touches most lives. I'm sure you have friends, family, even yourself that have been affected by cancer. The stats that I show here, this is, staggering numbers. 609,000 people will have cancer, will die from cancer this year. Not just have it, but die from it. And 1.9 million people will be diagnosed with cancer in the United States. So you put all these numbers together, and the probability of a man or a woman being diagnosed with cancer is a little more than 1 in 3 individuals. And think about how many people are currently listening to this. A significant number of you will be affected by cancer. So some stats that you may not be aware of, though, are these, because we all think that we've advanced, we've got new treatments coming down the road. Immunotherapy has been a huge, impact on cancer survivability. But if you start looking at the numbers here, 90% of cancer clinical trials are going to fail. And that's a staggering number. Even worse, there's about a 5% chance of success of any new cancer drugs that are being discovered in labs like mine, moving forward from preclinical to being approved by the FDA. The FDA is that big organization that approves all the drugs that go into treatments for people. And so those are pretty bad numbers. We're not doing well at all when it comes down to moving new discoveries in labs like mine into the hands of the cancer patients who so desperately need them. So why is that? What are the reasons why we have such poor success in moving new treatments forward? Well, maybe it's not intuitive, but basically every cancer patient is different. And hopefully I'll explain to you all the approach that the current field is using to develop and test new technologies is actually flawed, because it doesn't take this into consideration, that every cancer patient is going to be different. The bottom line is that they're not testing the right products with the right people. And I want you to keep this in mind as I give you my presentation today, because this is really the strategy that I'm taking in the lab to ensure that I don't fall into that 5%, that we're in that group that does, get through FDA approval, to make sure that when we get to the point of testing our products in patients, we're testing them with the right patients. So here's kind of, and you all may have heard of 23andMe and all the genetic testing that's going out there. So there's a lot of genetic testing that's being moved to do in the cancer field. So 23andMe, for example, can give you a cancer screen. You can buy the kit, throw your DNA in there and give it back to them, and they will give you all the genes that you may have, what's called the germline that you're born with, that might make you susceptible to cancer. There's also another host of tests, and that's the ones that Foundation One does. If you do happen to have a cancer and you get a biopsy, they will take that biopsy specimen from your tumor to Foundation One, and they will screen for any of the potential potential genes or mutations that developed over time to cause that cancer to occur. And the reason they're doing this is this idea. They have this idea that if they can identify what are the drivers, what caused the cancer in that patient, and this picture here kind of shows you different genes that got mutated in different cancers and individuals, they're going to want to match the mutation with a drug, and hopefully what that's called personalized medicine, to try to find the ideal medication treatment plan that's going to kill the tumor cells that are making your tumor grow. That's the current dogma. That's the idea that everybody's got. But there's a problem with this idea that as I said, I can't wrap my head around truthfully this personalized medicine approach, because I think it works for some individuals, but not for the vast majority. And here's why. This kind of shows you how cancers develop over time. And what you see here is, in the beginning, the gray cells are normal cells, non-cancerous cells. And over time in adults, you start to acquire different mutations in cells. And most of the time we eliminate those cells that get mutations. Our body's really good at getting rid of them. But as we age, things happen. And sometimes those mutations, those cells that have these mutations, don't go away. And over time, they develop different populations. So you can see here in the second group, we now have these pink and blue tumor cells. Well, the colors represent different mutations. And so you can see that over time, tumors become what we call heterogeneous. They're composed of not the same cell with the same mutations, but multiple cells with multiple different mutations. So if you go back and you try to screen and you do, let's say they take a tissue biopsy of this really large tumor that has all these different types of mutated cancer cells, and they take a piece of the tumor. What if they take a piece of the tumor over here where the purple cells are, all right? And they say, oh, you've got the purple cell mutation, so let's give you a drug to target the purple cell mutation. It's not going to affect the blue cells or the orange cells, because they didn't sample those cells when they did the biopsy. Or what happens over time is, maybe if they treated you when you only had the orange and blue mutations and you got a treatment for those, you never got a treatment for the purple cells, or the ones that maybe caused metastases or recurrence over time. So it's basically a moving target. And kind of this picture shows you that. We have differences between the tumors between patients. That's called interpatient heterogeneity. So patient one might have breast cancer with these mutations, and patient two will have breast cancer with different mutations. But even more, within the same patient, you'll find differences. So maybe a tumor that occurred in the breast will have a certain number of mutations, but when that tumor metastasized and maybe went to the lungs, it'll be different mutations. And again, trying to find the treatment is a moving target. And I hope that's kind of coming across in what I'm saying here. And let me stop for a second. Any questions so far? Ready with me still? Okay, and I'll keep going. So what are we doing? We've all, and many labs like mine think the same way. We've been thinking a lot about this, and how can we address this? And basically, what we're trying to do is called the paradigm shift. We're trying to kind of shift the thinking that people currently have with this personalized medicine approach, and try to develop therapies that inhibit something, and the something is what I'm going to talk to you about today, that cancer cells need and doesn't go away over time. That's really important. So the cancer cell is going to need this something that we're going to target therapeutically throughout its life of being a cancer from over the years, basically, that the cancer takes to develop. And even more so when it becomes metastatic. And a lot of our work that I'm going to show you today really is targeting the metastatic cancers, because there's nothing out there, truthfully, that's truly effective against metastatic cancers. So here's the idea. And I'm taking you guys all back to your high school biology. This is one of the central dogmas of biology. DNA is the blueprint. It has the blueprint for all the proteins, which are basically all the things that your cell needs to function, live, survive, grow. The blueprint for all the proteins that your cell makes is your DNA. And the RNA is kind of the translator. It translates the blueprint from the DNA into protein. And so what we want to do with our research is not focus so much on the mutations and the genes and the DNA changes that cause cancer, but rather on the proteins that result from these changes, the DNA, the mutations, and the genes. Focus on the proteins or the products and target those therapeutically. And again, we'll get to how we're going to do that in a second. So I show you, this is a very complex picture. And what this picture basically says, this is kind of called the hallmarks of cancer. And if you go to any cancer talk out there, people always refer to the hallmarks of cancer. And what these basically are the behaviors that cancer cells do that make them cancer cells. For example, growing uncontrollably, avoiding being killed, accessing more nutrients, that's basically what all these things cause. Inflammation. All these things are behaviors that cancer cells do that makes them cancer cells. And all these behaviors are not caused by the genes. They're caused by the proteins that the genes encode. So again, we're getting back to the proteins being the critical piece that's causing all these changes in cancer cells. So this is kind of my own little rendition of this idea. So just I'll walk you through this in a moment. What the current idea is that we're going to identify all these genes. And I just told you that, they do Foundation 1, they do 23andMe, whatever they use. They're going to try to identify the genes that are mutated in a cancer. And then they're going to try to find a drug that's going to target the protein that's made by these mutated genes. That's the current idea. We're coming with something completely different. What we want to do is shown in this picture. There's actually a connection between the genes and the proteins that cause cancer. And that's called the chaperones. And what chaperones are is that they are these, think of like little machines that will actually fold proteins. And why do we need to fold a protein? Well, when a protein is first made from the DNA blueprint, it has no shape. It's called unfolded. If you think about it like a long necklace of beads, it's just this long linear strand, has no shape, has no function. You need to actually fold it in the proper three-dimensional shape. And think about it like a lock and a key. You have to make the key fit the lock. You have to fold the protein in the right shape so it's going to have the right activity. And that produces a functional protein. And what's responsible for going from unfolded, which has no function, to folded that has function are these chaperones. And they're actually a bottleneck. So that's kind of why I drew this diagram showing you this piece, this point here, is actually the Achilles heel of the whole process of DNA to protein. It's the folding of proteins. And that's the point that we're targeting. We're targeting the ability of a cancer cell to fold proteins. And you can see that it's critical, because if you block this ability, everything stops right here. That cancer cell, it doesn't matter what mutations it made, it doesn't matter how it changed the DNA blueprint. If it can't fold the proteins that come from the DNA, that cancer cell's dead in the water and it can't proceed any further. It basically will die. And we'll show you that data. So that's where we're heading in our lab. So protein folding is actually done by an assortment of different components that every cell has. And this kind of picture shows you the three major classes of these protein folding machines. The heat shock proteins actually have been studied for many years. This is something that, at least for the last, I would say, 10 to 12 years, people have been trying to develop inhibitors to block the heat shock proteins. The problem is the heat shock proteins are among the most abundant proteins in normal healthy cells. So trying to knock this down in a cancer cell is going to affect a normal cell. And that's a major problem with the heat shock protein. So they're really not a good target for therapy, at least for cancer therapy. We're focusing on a different type of protein folding complex. It's called the chaperonin. And a chaperonin, if you think about it, it's like, it's a large multi-protein complex, and it's composed of two discs, almost like two little donuts that sit one on top of each other. That's a picture shown here. And it's found in both bacteria, and that's called GROEL, but the human version of it is called CCT, or chaperonin containing TCP1. And that's what we're studying in the lab. And the reason I'll show you in a second is that chaperonins are actually very specialized, and normal cells don't really need them very much, but cancer cells need them a lot. And that's what our lab's been proving in the last few years since, we've been working with the Orlando Sports Foundation. So here's our target. It's called chaperonin containing TCP1. As I said, it's large, megadalt, it's huge. It's a very large complex, multi-subunit, 8 different components that come together in this two-ring structure. And if you look at it from the top, it has this hole in the center, and actually the hole opens and closes. So when it's functioning, proteins go inside of it. That linear, necklace-like no-shaped protein goes inside of it. This hole will actually close, and that energy from closing the hole actually powers the whole complex so it can fold proteins. And when it folds the protein, it reopens, and the now nicely shaped, functional protein pops out. And here you can see the side view. And I'm showing you the side view here because one of the first challenges that we have studying this very large complex unit is that it's made of 8 different components. Do we have to study all 8 components at the same time, which is a huge undertaking? And what we first did is try to figure out, is there one piece of this that's really, really important? And we did, and we found that this one piece called CCE2 is actually very important. And we can just study this one piece. Instead of studying 8 pieces, we can study one piece and show that it reflects what the rest of the other subunits are doing, which was a very powerful finding. Okay, again, stopping it quickly if there's any questions at this point. Okay, if not, I will keep going. So,
Annette, you're doing a great job. Thank you.
Oh, sure. No, enjoying everybody.
Cool.
Hopefully someday we can do these back in person, just because it's nice to see people. But at this point, I am very happy to do this from my office by Zoom. But this is an older piece of data. I'm showing you this because this is where we started. And you can see the date here, 2016. This is when we first got funded by the Breast Cancer Research Foundation. And we had just discovered that this chaperonin that I'm been talking about was highly elevated. Its expression was highly elevated in breast tumor tissues. Like I said, it's older data, but it kind of shows you where we started. And what we found is shown here in this little graph. Cat is not a cat. This is called cancer-adjacent tumor. And what this is normal, healthy tissue that's just next to the tumor, but it's not tumor tissue. And you can see that the levels of our chaperonin were very low in normal tissue. But when we looked at what's called invasive ductal carcinoma, IDC, which is the really invasive, very aggressive form of breast cancer, we found that it was very elevated, and it actually went up the more advanced the cancer was. So T1, T2 was like an early stage of this invasive form of cancer, and T3, T4 is a later stage, almost going to metastases now of this form of cancer. And we saw this pattern of very, very low in normal healthy tissue, high in the tumor tissue, and higher in the metastatic, more aggressive tumor tissues. And this data we show you here for breast cancer, we found the same pattern in lung cancer, in prostate cancer, in colon cancer, and I'll show you today a little bit more in pediatric cancers as well. So adult and pediatric cancer, similar pattern, which is showing you that this chaperonin is needed by cancer cells under all these conditions, which fits our hypothesis, which was that this is going to be like a universal need that all cancer cells need. So before I keep going, this is I'm very passionate about training and mentoring in my lab. And while we're doing all this research, we're also training the next generation of scientists. So I also wanted to update you all that my 3 superstars, which had done the bulk of the work in the last 3 to 4 years, have all graduated. They've all gone off to do better things, which is what they want to do. So Amanda just graduated last summer, and she's now at medical school at UCF getting her MD. She's going to be an MD PhD scientist. Heba is now also graduated from my lab, and she's now beginning her own lab. She's actually at Jordan University in her home country of Jordan, and we've been Zooming, almost weekly, continuing the work that she's carried on, and now her new lab herself that she's going to be beginning. And then Daniel was also recently graduated. He is now a postdoctoral scientist working on creating body on a chip. So this is a new technology where we can test drugs like our inhibitor that we're testing for the chaperonin, instead of having to use mice or basically living organisms. We can do the testing on a little microscope side, because they reproduce all the body's organs on this little microscope slide. So that's what he's been working on. So I just want to share that, in addition to research, we also are training the next group of scientists. So my new team, I don't have pictures of them yet, but I do have a new team in the lab, and they're all just beginning their careers. James is getting his bachelor's degree at UCF. Colton and Carolyn are getting their master's degree, and Lamb, like James, is also getting her bachelor's degree. So we're doing that next round of new folks that are training in the lab. Hopefully this will be our next group of superstars that are doing the research. But back to our chaperone. So one of the biggest challenges that we had starting in 2016 is shown kind of in this slide. This is a slide of all the publications, all the scientific publications that have come out since 1994 that report something about the chaperonin and cancer. And you can see that really through 2016, it's 2019, there were very few publications in the entire world. So in the entire world, there are about 4 to 5, 2 to 3 publications every year on CCT and cancer. So it was not studied by anybody, truthfully. Very, very few publications. And really, there were no what I call longitudinal studies, like one lab publishing regularly every year. There are no labs. These are all like one-time publications. We are probably one of the only labs, if not the only lab, that has published continually, and my publications are shown here in the red. Since 2016, we've been publishing 1 to 2 papers almost every year showing how the chaperonin works in cancer. And it's having an impact, because you can see now that starting in 2020, the publications are jumping. More people are now understanding and appreciating how important this chaperone is in cancer. But we got a head start, because we started before everybody else did, publishing and working with the chaperonin. So what we did, again, what we have published in the last year, we've published 3 papers in the last year and a half. And the most recent paper was our neuroblastoma paper. And this is where I'll talk a little bit about that. We reported that the chaperone is very critical for pediatric cancers. And this is the first report of anything to do with protein folding in a pediatric cancer. As I said, it's had already 1,200 views, and it's only been out for a few months. So this is really encouraging. We also published a scholarly review. So what a review article is where you study the entire field, and you go in what everybody's published, and you summarize all those discoveries of 20 or 30 years of research in one paper. And that's what I did here. And that paper has been extremely well received. Again, just was published a few months ago, and already we've had 2,000 views. And this, I just learned a couple weeks ago, actually last week, that they've taken this review that we published, and now it's part of a chapter in a book. So the publishing company really liked it, and now they have it in a book form as well. So we're really excited about that. But the one that really has given us the most press has been this last one, which is our discovery of using the chaperonin as a diagnostic marker. And that was 7,300 views so far. And we've also had a lot of press. These are all different online articles that came out in the last year talking about our discovery. So it's got a lot of press also in the community. So we're very excited about our new liquid biopsy approach. So now that I've shown you kind of what we've done and what the press and the publications look like, let me talk a little bit about the science. What does this all mean? What did we actually publish? So one of the major discoveries that we've made in the last few years is that CCT, the chaperonin, is very important in causing a tumor to grow from a more like benign state to become very invasive and aggressive. And that kind of shows you, again, that little picture I showed you earlier, where you see the initial normal cells with 1 or 2 tumor cells, that over time, more tumor cells mutate, grow, and you get these very large, very heterogeneous tumors over time. Well, what we found that if you actually take these tumor cells, either non-tumor cells or very early stage tumor cells, and give them more chaperonin, so artificially add more chaperone to these cells, you can immediately move them from the either the non-cancer or benign shape or form to a very aggressive invasive tumor. So you bypass all those other steps. It goes immediately to an aggressive invasive form of cancer. And it happens immediately. We were able to take, imagine these gray cells and just give them a little bit more chaperone. Within a couple of days, they were acting like the purple cells, like aggressive, nasty tumor cells. So it tells you it's very, very critical for that process of becoming invasive and aggressive and metastatic. We also learned the other way around. What if you took away the chaperone? So you took an aggressive invasive tumor and you took away the chaperone, what happens? Well, it goes back. So what we found was if you take this very aggressive, nasty tumor and you got rid of the chaperonin, most of the tumor cells will die, but the normal cells don't. And so you end up with what you started with the normal or benign, tumor cells. So this is really important. Again, showing you the important role that the chaperone has in causing cancer. So remember when I talked a few slides back and I showed you all the different types of protein folding complexes that are out there, I talked about the heat shock proteins, and I told you the big problem with targeting heat shock proteins is that their normal cells need them, right? Well, do normal cells need our chaperone? Do they need CCT? And the answer is they do, but not a whole lot. So what we've learned, not me, my lab, but other labs who've been studying the function of the chaperonins in normal cells, is that it's a very specialized complex. It folds only a certain number of proteins under very specialized conditions in normal cells. One of the things that it folds are called actin and tubulin, and actin and tubulin are kind of like the bones of cells. And I'm kind of showing you this picture here. Green is tubulin, and red is actin. But cells don't need to make actin and tubulin all the time. They just need it once in a while, but not all the time. So what people have come to conclude is that about 1% of all the proteins that a cell makes, they need the chaperone for folding. The other 99% of the proteins don't need the chaperone. They get perfectly fine, without it. So that's very important. So we know that if we were to inhibit the chaperonin, we're probably not going to cause too much damage to a normal cell. It'll cause a lot of damage, hopefully, to a cancer cell, but not so much to a normal cell. So why? Why would it cause damage to a tumor cell? Well, this is what we learned. This is a very complex picture. You don't have to worry about the details. This just shows you all the pathways that are turned on in a cancer cell, all the proteins that get turned on in a cancer cell that cause cancer. And every single one of those is folded by the chaperonin. So all these proteins that cause cancer, the chaperone is essential for folding them. And so we've kind of done the numbers and we've crunched the numbers with our data. And we would say that about 20%, at least 20% of the proteins in a cancer cell are folded by the chaperone compared to 1% in a normal cell. So the difference between 1% and 20% is actually huge. And why that's important, because it gives it what's called a therapeutic window. And what you can see here is that a CCT inhibitor, the dose that we pick, to give to a patient, can actually fall within this therapeutic window, where it's going to be a dose that kills the cancer cells that are highly dependent on the chaperonin, but it's not going to get to the point of the red, which is where you now start having damage to normal tissue. So the therapeutic window is a window of dosing where you can have the most effect on a cancer cell, but not have any effect on normal tissue. And because we show this difference between 1%, 20% in the proteins that are affected by the chaperone, we probably will have a therapeutic window for targeting. But if you guys remember, I showed you a slide in the beginning that said that only like 5% of, things like a chaperone inhibitor actually reach FDA approval. And kind of the reason is that they're not testing the right product with the right people. So that is the first thing that we want to address. Before we actually get too far into developing chaperone inhibitor as a therapy, we want to make sure we have a way to identify the best patients to test the chaperone inhibitor. And the way we're going to do that is we're going to develop a diagnostic. So that's what I'm going to talk to you a little bit about right now. We have spent the last year really working hard on advancing a diagnostic assay that's based on detecting that subunit 2 that I mentioned. It's really important. And we had several choices of diagnostics that we could develop. We could either develop what's called a standard biopsy diagnostic, which is basically relying on doing some sort of needle biopsy where they take a piece of tissue from a tumor and they send it to a pathology lab, and a pathology lab would then test, for example, for the presence of the chaperone. But the problem with this approach is that you have to already have had a cancer, and it's really not very good for doing this frequently. This tissue can cause, the process is invasive. It can cause damage. There is pain and risk associated with any type of biopsy, and you really can't do it regularly. We really wanted a diagnostic assay that could be done easily, regularly, and can be used for patients, throughout their whole time of being treated with a drug or a treatment. And so that's where liquid biopsy comes in. Liquid biopsy involves drawing blood, urine, any sort of biofluid, and analyzing that material for the presence of tumor something, right? And it's good because, as I said, it's part of standard procedures. You get your blood drawn all the time. It's something that can be done quickly, easily, and it's a lot less invasive than a tissue biopsy. So we first started to look at what's called circulating tumor cells. And what circulating tumor cells are, here you see there's a primary tumor that's growing like a breast, for example, a breast tumor. And those breast tumors have blood vessels that are attached to the tumor. And what the tumors do is they actually will shed pieces, little cells, from the main tumor into the blood, and that those cells will circulate through the body. They're a very small number. They're not the most abundant cell, obviously, in the blood. There's many other cells present in the blood. So they're rare cells, but they are found in the blood. And they're called circulating tumor cells. And it's thought that these are the cells that are responsible for causing metastases. So it's a really important cell to detect, because if you detect a lot of these cells, that's a warning that the patient may actually be either metastatic or going to be metastatic in the near future. So we decided to design a diagnostic assay that uses the chaperonin to detect circulating tumor cells. So here's there's only actually one FDA-approved method or diagnostic for detecting circulating tumor cells in blood. It's called the cell search system. So we use that platform of the cell search system to test out our idea that the chaperonin could actually help us detect circulating tumor cells. So this shows you, this is from a study that we did with small cell lung cancer patients. We had 4 patients, and we used first the standard method, which is the currently FDA approved cell search method. And I just want to draw your attention to the second row here. This system actually lets you look at each circulating tumor cell from the blood, one cell at a time. And what you can see here of these 4 patients, if this was going to be the diagnosis, patient number 1, there was no circulating tumor cells detected. So this patient they would have said, oh, this patient doesn't have any cancer because there's no circulating tumor cells present. Patient 2, maybe they would have said, yes, possibly here's 1 cell, but it's very faint. And patient 3 and 4, again, maybe, but it's kind of faint too. So really, none of these 4 patients would have actually been a solid positive because of the fact that they're really not very strong. So the standard criteria, and it's known that it does this. It actually, this method, the cell search method, is actually not being used very much anymore because of this problem. It actually misses a lot of the circulating tumor cells. Well, we did it, but instead of using the standard method, we used our chaperonin to identify circulating tumor cells. And again, if you look at that second column again, you can see here. So we did find in patient 1 some circulating tumor cells that were missed with the standard protocol, as well as patient 2, 3, and 4 all had circulating tumor cells that we could detect with our chaperone. And this finding was that paper that I showed you a few slides back that had over the 7,000 views and got all the press in the field, was this discovery. And so this has been a really strong point that now we can use this method of this diagnostic. Once we validate it, which I'm going to talk about in a second, we could theoretically use it to screen cancer patients for future clinical trials of a chaperonin inhibitor. And that's where we're heading with this. So let me tell you a little bit more about, that was adult cancers. We actually, in the last year, also worked on pediatric cancers. And the reason we did pediatric cancers, because adult cancers, there actually are some diagnostics out there. There's, I told you, the cell search system. There's, there's mammograms for breast cancer. There's colonoscopies. There's tests out there to help detect adult cancer. There are no tests for pediatric cancers. Pediatric cancers are actually a leading cause of death in children and teens. And we don't know what causes pediatric cancers. It's not the same thing as adult cancers. There's no genes. It's not like with the adult cancers, we can do all these genetic testing. There's no genes that we know of these pediatric cancers. So really, it's an area that is completely needy for new diagnostics. And this is just showing you what we first tried to look at. Okay, is the chaperonin present in pediatric cancers? And the answer is yes, a whopping yes, because it's actually present at even higher levels than adult cancers. So it's very highly expressed in pediatric cancers. And again, getting back to the idea, this is actually a universal marker. It's not tied to whether I have breast cancer or I have prostate cancer or I have lung cancer. It is, do I have cancer, period. It is a universal marker of cancer. So it has applications that can go, as I said, now adult, and now I'm going to show you data that it also works in pediatric cancers. So I'm not going to give you all the details because we talked about a little bit this last year, but the cancer we chose was neuroblastoma. And the main reason is actually point number 3. Neuroblastoma is not a brain tumor. It's actually a cancer of the nerves, and it's frequently found in the abdomen, but it's basically diagnosed in children under 1 year of age. If you think about a baby, these are the infants are the ones that get neuroblastoma. And the challenge is that if they happen to detect the neuroblastoma in a 6-month-old or 8-month-old baby, doctors can't do anything at that stage because a lot of times those neuroblastomas actually spontaneously regress. So basically the doctor has to wait to see if that neuroblastoma in that infant actually gets worse or gets better because they can't do anything when they first detect it. So it's really a challenge. And if you wait till it gets worse, it's even harder to treat. So this is a really serious cancer, and having a diagnostic that could help a doctor make that decision when they first, find the neuroblastoma in the infant would certainly help improve the survival of infants that have this type of cancer. And this is just showing you what we found. Again, this is a biopsy. Now, this is a tissue biopsy, but we found that this is the normal tissue on top, and the darker the brown, the more chaperonin is present. So you can see here the normal tissue has very, very light brown, which tells us that normal nerves don't make a lot of chaperonin. But once you start looking at stage 1 or stage 2 neuroblastoma, you can see how it gets really, really dark brown. That's super high levels of the chaperonin that are found in these neuroblastoma tissues. And what was really important is I want to show you these first two on the second row. You can see this very first panel looks really light, not very dark at all. Although it's neuroblastoma, it's stage 1, but it actually didn't have any of the markers. So down here, you see where it says C56 and CGA. These are markers for neuroblastoma. This tissue did not have any markers for neuroblastoma, although it was considered neuroblastoma. It's probably one of the ones that just regresses. And you can see that we saw no chaperone in that tissue. But all the other ones that had very high, you can see the pluses, these are all the markers for neuroblastoma, coincide very strongly with the presence of the chaperone. So again, this data just helped to emphasize that this could be a very good diagnostic marker for neuroblastoma. Won't belabor the point, just we found very much like we saw with breast cancer, that if the cells express very high levels, as a matter of fact, the levels that neuroblastoma expresses of the chaperone are as high as metastatic breast cancer, so they're very high levels. We found that if we added more CCT, these neuroblastoma cells became more aggressive. If we took away CCT, these cells died. Where was really the critical piece here was that we could show that we could use that cell search diagnostic to detect circulating tumor cells now for neuroblastoma. So the idea is that you would basically have an infant, you can draw a little bit of blood from that infant, run it through this diagnostic, and if we find circulating tumor cells that have the chaperone, that would alert the doctor that this child's tumor is getting worse. And what's really important is that the cell search system cannot be used at all for neuroblastoma. This first panel shows you if we were to use just the standard criteria for cell search, everything is blank. It cannot detect a single neuroblastoma cell. But if we use our chaperonin to detect, even using that same cell search system, but switching it out for the chaperonin as a detecting mode, you can see we can detect lots of neuroblastoma cells. had a question. I think, Robert, you had a question for me?
Yes, can you hear me?
Yes.
Hey, Dr. Khaled, thank you. Once you have like one marker, like, is there a magnitude of like when you have that marker, does it mean that you're automatically going to be prone to having the growth of a tumor or cancer, or are there kind of like false positives with markers?
That's a great point. And I'll tell you the theory, and then I'll tell you what we're going to do next to determine whether there are false positives. The theory is that yes, any level of chaperone, and kind of I can show you the picture here, you can see that there's different levels of staining. You can see some that are really, really strong and some that are a little bit lighter. But any presence, if we're correct with our theory, any presence of chaperone is actually a sign that the tumor is getting metastatic and it's becoming more aggressive. Now, to do false positives, we actually need to have a much larger pool of patients to study. And that's where we want to go. I'm going to show you what we're going to do next to actually address that point about having those false positives. This is where and then your question actually set me up perfectly for my next slide. What we want to do is we want to validate this new CCT liquid biopsy that we've now shown works in breast cancer, lung cancer, and also pediatric cancers. We want to validate in a large patient cohort. And we've actually chosen prostate cancer to do that with. And the reason is prostate cancer is a cancer that takes many years to develop. So if you guys get your PSA test, and if you happen unfortunately to have a positive PSA test, they'll do a biopsy. But usually prostate cancer caught early, surgery is very effective, radiation therapy is very effective, and usually that's enough to treat the cancer because prostate cancer progresses very slowly. It's a 15 to 20-year progression of prostate cancer. But unfortunately, for those patients who do progress and develop the metastatic form of prostate cancer, it's very challenging because there are no treatments that really are effective. There's no plan for how to handle metastatic prostate cancer. And importantly, there's no diagnostics because the PSA does not work once you're metastatic. PSA is not cancer specific, it's prostate specific. So it's not very good at detecting the difference between a benign cancer or something else that causes some abnormality in the prostate. So there's nothing out there to detect and diagnose and guide treatments for metastatic prostate cancer. Why the chaperone will be very good as a diagnostic for prostate cancer is shown on this slide. So they've already identified 3 main gene products that drive from low risk to high risk and metastatic prostate cancer. And that's MYC, the antigen receptor, and beta-catenin. Those 3 gene products are drivers of metastatic prostate cancer. And all those 3 are folded by the chaperone. So we did a big, what's called a bioinformatics study. This is basically data mining, all the data that's out there, thousands and thousands and thousands of data sets that are out there. And we found that the chaperone levels are very high as you move from low risk to high risk to macrometastasis, which is not a metastatic form of prostate cancer. So there's really good reason to study this in prostate cancer. Where are we going to get our patients? How are we going to have a big cohort of patients to study our diagnostic and do exactly what Robert just asked us about, finding out, is there a threshold? Do we get false positives? Do we have false negatives? To try to do that, we need a large cohort. Well, our Orlando VA actually has one of the largest populations of prostate cancer patients in the veterans that they see. And this table is showing you all the numbers of metastatic and prostate cancer across the country at all the VAs. Our VA here in Orlando is part of what's called the VISN 8, which is the Veterans Integrated Service Network. The federal government loves all these anagrams, but number 8 is where Orlando VA is. You can see that they see something around 12,000 metastatic prostate cancer patients. That's a huge patient population to do our study in. If we could study these patients, we would be able to validate and move our diagnostic to the next level, which is actually being used in patients. So these types of studies cost a lot of money. And how are we going to get that money? That's the first question. So there are 2 main sources for funding for labs like mine to do these large-scale million-dollar studies. It's either NIH, which is the National Institutes of Health, or DoD, the Department of Health. And both those have both what's called extramural and intramural. So extramural means anybody who's outside the organization. So for example, I'm extramural to NIH and DoD. They also have funding for intramural, which are their own scientists, like NIH scientists, which I used to be one many years ago, and DoD scientists. But I'm an extramural, so I have to apply to extramural grants. And I will tell you the funding success of these two organizations is less than 10%. That means that out of every 10 grants they get, they fund one or less. So it's extremely, very difficult and very competitive to get grants from these organizations. But there's one more organization, which is the VA itself. Actually, VA, like NIH and DoD, has a very robust grant funding mechanism that will give you grants to do these types of large-scale clinical studies, which is what I'm proposing to do. But there's a caveat. It only is intramural. So only people who have VA, who are VA scientists or VA employees can apply for VA grants. But there is a loophole. Being an extramural person like myself, I can apply to the VA to become an intramural scientist. It's a very difficult process. It's very stringent. They accept, again, less than 1% of the people who apply get accepted into this program to be intramural. And I was very, very lucky because I was accepted. So I currently have intramural status at the Orlando VA. So I can submit VA grants. So we used a lot of the support that we got from the Orlando Sports Foundation last year to generate the preliminary data to submit a VA merit grant application. And this grant was for $1.2 million. We just submitted it now in March, this month, early this month, and we're keeping our fingers crossed. The next steps is this grant is going to be peer reviewed by a VA study section. It's going to happen sometime in May. It's then going to undergo the programmatic funding review by the same people, by the way, who gave me the intramural status, the same VA Central Research Office. So I'll find out sometime in July whether we got funded or not. I will tell you, it is still a stringent process. They fund about 20% of the grants they get. So again, 2 out of every 10 grants that get submitted get funded. So more than likely, I'll have to go through several rounds of this process of, addressing reviewers' comments, resubmitting the application. But I have 3 tries, so that when they accepted me as an intramural scientist, they gave me 3 opportunities to submit a grant. So we just used one of them right now to submit the first grant. So like I said, we'll go through this process until we get it funded. If, however, we are lucky and we get funded the first time, which I say, miracles could happen, things could happen, we would get the grant awarded, we probably will get the dollars to start that large-scale clinical trial of our diagnostic sometime about March of next year. And that's when that project will begin. But as I said, it is thanks to the support that we get from the Orlando Sports Foundation that we were able to actually move to this next step of testing our diagnostic in now a large patient population and have an impact. Because if what we show is true, that we actually have a diagnostic for metastatic cancers and we show it with these veterans, we're not only helping the general population, we're helping our military veterans, which really deserve our care and attention as well. All right. So any other questions here before I continue? All right, so I'm going to take the last couple of minutes to tell you the other big news that we have, which is now, that was all the diagnostic, now is our therapeutic. So our therapeutic actually has been around for a number of years. We actually discovered this it's a small 20 amino acid peptide we call CT20. We discovered it about 10 years ago. We knew that it killed cancer cells 10 years ago, but we had no idea how it worked. We just knew that it killed cancer cells. And it shows you how long science takes. It's taken us a period of, let's say, almost going on 10 years of trying by trial and error to discover bit by bit what its activity was. We first, the delivery system that we were using, that we have been using the last 10 years to deliver the peptide to cancer cells, has been using nanoparticles. And they're made with this polyester polymer, and it basically, it will encapsulate free peptide. So basically the peptide works its way into all these little pockets that the polymer makes, like a sponge, think of like a big sponge with pockets, and each little pocket will contain the peptide. And cancer cells will take up the nanoparticle inside and then release the peptide into the cell to eliminate or block the activity of the chaperone. So our CT20 is our chaperone inhibitor. And this is an old slide, but it kind of shows you how effective it is. This is actually colon cancer cells, and you can see here how they're all just dying. as I said, we discovered that it actually, the way it works is by eliminating the chaperonin. And we recently found that the way it works is it actually blocks the energy part of the chaperone. So the chaperone needs energy to fold proteins. We found that CT20 actually binds to the site where the chaperone needs energy. So it prevents the chaperone from functioning by blocking energy. So here's the last couple of slides. I'm almost done. We were at the point last year that we were trying to figure out what to do next. So do we continue with the nanoparticle as our delivery vehicle? Or do we develop a small molecule that's a mimic of this peptide? Or do we develop a new system based on gene therapy? And we actually opted for the gene therapy. The problem is that the nanoparticles, no matter how well we optimize the targeting, most of it ends up in the liver. About 80 and 90% of the nanoparticles with CT20 will end up in the liver, and only a very small amount actually reaches a tumor. That's not practical to go into human studies with that very low efficiency. Small molecules, I actually talked to a bunch of folks. We actually went out to Alabama to go talk to Southern Research Institute, that's a huge small molecule organization. And we all came to the same conclusion. It's going to be very hard to mimic a small molecule to do the exact same thing our peptide does. So small molecules kind of out, which really left us with the gene therapy approach. And that's where we're going to head next. We're going to try to develop a method to express CT20, our peptide, attached to some sort of targeting protein, and put it in some sort of genetic vector so that the cancer cells will make the CT20. We don't have to give them the peptide anymore. We'll actually give them the gene to make CT20. And hopefully once the cancer cells start making CT20, that will kill the cancer cell. What we're going to pick as our gene delivery system is actually a bacterial vector. We're working on developing salmonella. You all go, food poisoning, salmonella. Well, actually, salmonella has been FDA approved to treat cancer patients because salmonella naturally targets tumors. So unlike, as I said, the nanoparticles, where only 10% of them actually reach the tumor, 100% of salmonella reach tumors. So much more efficient delivery system, already FDA approved. So we're working on developing this. And we were just funded by the Little Like Bella Foundation to develop the salmonella-based CT20 CCT inhibitor for neuroblastoma. So we're really excited about that. So I just want to remind you all as I finish up what we did this year. New liquid biopsy, submitted the VA grant to start large-scale studies of that. New delivery system based on salmonella, new grant to move that forward. And we had 3 students graduate. What we're doing next is we're actually going to the next generation now, the diagnostic. We're going to get out of the cell search system and develop what's called an exosome system, because this system is actually a lot cheaper to run. The cell search system is actually very expensive. So we want to have a cheaper, more efficient system of detecting our CCT in tumor cells. So that's what we're working on. We're going to hopefully keep that VA round going until we get funded. And as I said, developing the new salmonella system with our new funding. That's it. I thank everybody for their attention. And if there's a few minutes left, I'm happy to answer any more questions. And let me stop
We
here.
definitely have time for that, Annette. And I will close, but I want to make sure everybody has an opportunity to ask Annette a question here. That's the reason for the Zoom today. Any other questions at all or comments? Feel free to comment.
And thank you all for the good wishes in the chat.
Absolutely. Absolutely. Well, congratulations again on Annette on the publishing. I think we all understand the importance of publishing and for it being received by the cancer research world in general. So congratulations on that. And I just want to thank everybody here, for being a part of the Orlando Sports Foundation. You can take honor, in being a part of that, because this stuff does not happen without the support of this organization, as Annette is said, and is always so appreciative of it. And we appreciate her coming out when she has time. We don't want her to pull out of the lab too much, because we don't want to keep her off track of the goal, and that is to solve cancer. And so we certainly appreciate when she does take time away from the lab to give us some update. And it's certainly motivating for us, our staff, our stakeholders, Annette, when we get these updates, because it is the integrity of everything that we're doing. you know, in the Orlando Sports Foundation, we have 10 plus events that we do in support of cancer research. Obviously, our signature event is the ESPN event, Cure Bowl, which is ESPN has just done an amazing job giving us an opportunity to promote at even a higher level by giving us matchups that include top 25 teams, which they've been able to do the last 3 years. So we're very grateful for that opportunity, and obviously all the title sponsors that have been on board, from Auto Nation on down to help us launch. It's just been so important for us to do that. As a reminder, we also have the Orlando Sports Foundation also has a couple golf tournaments, a bowling event, our race. All of those events go to bring awareness to what we're doing and promote the signature event, obviously the Cure Bowl, but also to raise money for Dr. Khaled and her team's cancer research. So we're going at it. This is 15 years of the Orlando Sports Foundation, and we've hung true to our mission statement, which is bringing teams together to find a cure for cancer. And obviously that includes a whole lot of different teams and a lot of different events and sports, as well as the health field. And so we just couldn't be happier about the direction that we're going. And it's always an honor to see you, Annette, and know that we're helping you reach your goal, which is to solve cancer. You know, Matt Stover, who you see here as our associate director, I can't say enough about Matt's work. You know, he's been with us since day one, so this will be his 9th Cure Bowl. But we were talking about Cure Bowl, and a lot of times when we're all out there in the community trying to get people involved in our events, we often hear it a lot of times. Well, who's playing in the Cure Bowl? And Matt said, well, you know, ultimately who's playing in the Cure Bowl? It's Orlando versus cancer. And so again, it doesn't matter who's playing in the game, who's coming to the events. You know, we need people to do it because we want Orlando to help and be a part of solving cancer. And we know that Dr. Annette Khaled is our shining star to be able to do that. So it's an honor to work with you, Annette. Honor.
Well, I love being part of all of this. So thank you for the opportunity. I appreciate it.
All right. Now, it would be remiss if I didn't mention our next event is the Cure Cup. We're just about sold out. And that'll be out at Alworth this year on Monday, April 24th. But then please, you'll see my note in the chat. Please go to orlandosportsfoundation.org to see the rest of our events. And then obviously our curebowl.com for everything that is Cure Bowl. And you'll see all of the updates that we have going there. But we look forward to seeing everybody that is going to be a part of our Cure Cup out at Alworth. We've got a special check presentation with the Florida Breast Cancer Foundation to Annette. And so we're looking forward to that as well. So thank you again, everyone. We really appreciate you being a part of the Orlando Sports Foundation. And let's go Cure Bowl. Thank you.